Diabetes is a disease which affects millions of people in the modern world. It is caused by either no insulin production or a cell's resistance to insulin. Insulin is a protein-based hormone produced by beta cells in the pancreas. It is first formed as pre-proinsulin, then converted into proinsulin, and finally insulin. Insulin and glucagon are in charge of regulating the body's blood sugar levels. In a person without diabetes, when the body's blood sugar levels become too high, the beta cells in the pancreas release insulin, which signals the body cells to uptake the glucose. Let's take a closer look within the cell membrane to see how insulin signals the uptake of glucose. When insulin binds to an insulin receptor, it activates tyrosine kinase. The activation of the tyrosine kinase domain phosphorylates the insulin receptor substrate and sets off a chain of protein signaling. Along this path, a signaling protein phosphorylates a residue on AKT2 and fully activates it. AKT2 signals the phosphorylation of another molecule to regulate the transport of GLUT4 vesicles to the cell surface, thus allowing the uptake of glucose. After the cell has absorbed glucose, the liver then takes up any remaining glucose and stores it as glycogen, bringing the glucose levels down to a healthy level. When blood glucose levels drop, alpha cells in the pancreas stimulate the release of glucagon into the blood. The liver breaks down glycogen and releases glucose into the bloodstream, bringing sugar levels up to a healthy level. Type 1 diabetes is caused by an autoimmune disorder which causes the body to attack its own pancreatic beta cells. Thus, the pancreas cells cannot produce insulin. In type 2 diabetes, the pancreas does produce insulin, but the cells are resistant to the insulin. Let's take a closer look to see how this resistance occurs. The insulin resistance is the outcome of a problem with the molecule AKT. The insulin molecule binds to its receptor, but the AKT molecule does not phosphorylate, thus cannot signal the relay protein to open up the channels in the cell membrane that are meant to transport the glucose. Insulin resistance occurs mainly in obese people because there is so much sugar in their blood that insulin is constantly being sent to the cells, which starts to ignore the insulin. Insulin resistance reflects problems of cell signaling. Hyperproinsulinemia of type 2 diabetes is a result of a genetic mutation in DNA that codes for histidine at position 65 in between the A and C chain of proinsulin. At position 65, people typically have an arginine linking the A and C chain, but this mutation replaces arginine with the histidine. Therefore, the enzyme responsible for cutting off the C chain at arginine 65 can no longer do its job because it does not react with histidine. Although the C chain is still attached to the A chain, when a blood test is conducted on the patient, the proinsulin will show up as insulin. Yet, at the molecular level, the proinsulin could not react with the insulin receptor because it still has a C chain attached. The best possible treatment for this patient would be insulin therapy. Diabetes highlights the importance of cell signaling and the hormones such as insulin and glycogen in maintaining homeostasis.